Hello, Science 10, and welcome to our video on microscope calculations. This is lesson 2, and today you're trying to understand how imaging technology was created so we could better study the cell, which we just learned about in cell theory. So today, my goal for you is to understand the parts of the microscope, understand how microscopes help us see different sizes of cells, demonstrate how to use a microscope. Hopefully we can do that when we get back. Trace changes that have occurred to microscopes, and then do some microscope calculations. And then in another video, we're going to talk about gram staining and the key structures that deal with gram stain. So the four concepts in this video today are microscope magnifications, field of view, fit number, actual size, and drawing size. Okay, so there's four concepts, even though there's five numbers, just because two of them go together. But before we get into these, let's talk a little bit about reviewing, because you guys talked about microscopes in grade 8. So hopefully you have seen a microscope and used a microscope before, but let's just run through and in our notes label each of the structures. So structure number 1 is called the arm. Structure number 2 is the adjustment knobs. Now, there are two types of adjustment knobs listed here. I don't have them both separated out, but one, the inner knob, is known as the fine adjustment, which is the smaller one. It does small adjustments as you are trying to look at a slide. The larger one is known as the coarse adjustment. And what it does is that moves that stage, which I'll show you in a minute, up and down so it gets closer to the lens. The fine adjustment then helps get rid of the blurriness. Number 3 is known as the base. Number 4 is the ocular eyepiece. And this is going to be very important as we talk about magnification calculations today. So remember what ocular eyepieces are. The nose piece is the part that rotates, that holds the objective lenses. Now, the objective lens come in low, medium, and high power. Again, objective lens is going to be super important to today's lesson. So please remember low, medium, high is objective lenses. Here we have our stage clips and our stage. Our stage clips are what holds down the slide, and the stage is what holds the slide, the slide essentially sits on. Okay, and the stage is the part that moves up and down from the coarse knob adjustment. Then we have the condenser and the diaphragm. You guys learned in grade 8 that it was just called the diaphragm, which is fine. Essentially, it tells us how much light we're allowing in to view the slide with. And then last but not least is the lamp. So if you could add these into your notes, that'd be really great, as we are going to talk and review about parts of microscope in a drawing quiz. Let's move on to microscopes through the years. So let's start off with who did what and what are the types of microscopes. So first, Robert Hooke was kind of the first guy with compound lens microscope, which essentially means he had two parts to look through, two ocular lenses to look through, and it didn't magnify very much. And then Van Leeuwenhoek came up with a better version, and it could magnify up to, I think, 500 times is what it said. So then that was kind of the start of the whole idea behind microscopes. And now we in our lab and what we use at school are called light microscopes. So light microscopes are the ones that you just labeled the diagram of, and they use to illuminate the slide. They're pretty inexpensive. Now, when I say inexpensive, that's in relative terms to how expensive microscopes can be. They're not cheap, but they're the cheapest kind of microscope you can get. They're relatively easy to use. And again, they can magnify up to about 400 times. Then we have something called the electron microscopes. So you guys learned that electrons are the parts that move around on an atom. Well, we can use those electrons and beam them through instead of light. So replace light with electrons. And we make something called a micrograph. A micrograph is like a photo or a digital image of the specimen. And it gives us a much, much, much better picture. 1,000 times better picture. Now, electron microscope is a general, broad term. It's broken into a couple different types. I'm going to talk about two of the more important types, transmission and scanning. Those are the ones you kind of need to know for Science 10. The other ones are listed in your notes, the confocal laser microscope and the scanning tunnel microscope. Both listed, read them over as they are part of the evolution of the microscope. But the types of electron microscopes that we're going to talk about are TEM, or transmission electron microscopes. And they kind of work like a slide projector, sending electrons through a sample, and that gives us a picture. Okay, so they're kind of like, if you think about my projector on my roof that sends light to my board, that's kind of the idea. It sends electrons through, and then we get a picture. This is about 10,000 to 100,000 times in magnification. A better version of that is known as the scanning electron microscope. So what it does, or SEM, is sweeps a beam of electrons over an object. Kind of think of like a 3D printer. Kind of like goes back and forth and back and forth and back and forth until you get a 3D image. Now this can magnify up to about 300 times. Okay. Now from here, there's the confocal laser microscope, invented in the 1960s. It's more of taking like 2D images and making 3D images, and it can magnify about 18,000 times. The best type of microscope you can get, though, is called a scanning tunnel microscope. And it was developed in the 80s. And what it does is it produces things like images of DNA, and it uses an electric probe to do that. Okay, so this can magnify up to like 1,000,000 times the scanning tunnel microscope. But for this purpose, you need to know about electron microscopes such as TEM and SEM. Now, to kind of show you an idea, TEM looks like this. So again, it kind of just looks like a sample on a slide. This is an example of frog's blood using TEM, okay? But a scanning electron microscope gives us something like this. This is human blood with red blood cells, white blood cells, platelets, okay? It's more of an image that pops off the picture because it's 3D, right? You can actually see the shapes and whatnot. So very cool. So let's define magnification. Oh, I almost forgot. I forgot about one more piece to this, microscopy. So I'm going to link a video in on microscopy. You are going to, it's not a video, it's actually an article, sorry. You're going to read the article about microscopy. Now, not to confuse you, I added it in even though microscopy is not a microscope. It is actually the next step in the world of microscopes. It is super cool and awesome. And the best part is it's relatively new. 2019 was when they first started publishing about it. Even though microscopy has been around for a long time, DNA microscopy, very, very cool. So read that article and then come back and watch the rest of this video. So magnification. Let's define magnification and kind of talk about it. So all cells come in different sizes and different shapes, but the thing is they are all small. Okay. So I have a couple different examples of different types of cells that we would maybe look at. Sorry. The one on the one side, the green ones, are what we would call root hairs. Okay. And this is hair cell is, I want to say 15 nanometers in length. Okay. And the one on the other side is red blood cells. And what you're looking at are the red circles, okay? Red blood cells are small. You can fit 125 red blood cells across the tip of a needle. So think of a needle that you use for sewing. The tip of that, you can fit 125 red blood cells across that. So that is so tiny. So think about how microscopes are allowing us to see, okay? Magnification, through magnification. So what is magnification? It's a measure of how much larger a microscope or lens is. It's how much larger they make the image appear, okay? So for instance, a light microscope, we can magnify up to 400 times its size. So if something is 1 millimeter wide in real life, we could make it up to 400 millimeters wide using a microscope image. So for our purpose at school, there's not a lot that we need that needs to be magnified any further than that. So that's why light microscopes work well for us. So let's talk about microscope magnification as a whole. So here we go. Microscope magnification refers to how much larger we can make a specimen using our eyes and a microscope. So it depends on the two lenses used in the microscope. So this is where that vocab is going to come back in. Our ocular lenses. So the parts that are our eyepieces, the part where our eyes actually look through, that has already in itself its own magnification of 10 times. Okay. Then you have the objective lenses, the ones that spin. All right. They usually are found at low power at 4 times magnification. Please add these into your notes. You have a space. Medium power at 10 times magnification, and a high power at 40 times magnification. All right. Now we need to know 410 and 40 because I told you light microscopes can magnify up to 400, and this is how we figured that out. So for total magnification of a microscope, we can use a calculation that it says magnification is our ocular lens times our objective lens. So that means on high power at 40 times, our ocular lens at 10 times, we can magnify something at 400 times magnification, right? So our definition of magnification is how much larger can we make a specimen using a microscope? Well, magnification would mean ocular times objective. All right, so let's move into the world of, microscope calculations with field of view. So field of view is the diameter of the circle that is viewed under a microscope, okay? Now, I have a little disclaimer for you. This may be a little bit confusing because normally when I teach it, we have microscopes in front of us. And what we do is we look through the ocular lenses, we take a clear ruler, and we measure the circle we can see. In the lab, you can find low power and medium power field of view without any calculations. Your ruler will look, you can look through your eyepiece and your ruler will show it no problem. Okay? High power is a different story. But for field of view for our purpose right here, for most light microscopes on low power, field of view can be found, okay, by using 10 times our eyepiece and 4 times our objective lens, okay? So we use our magnification and then we just measure. So at low power, we have 4 millimeters, okay? So in your notes, you have examples in the slide. So what I want you to do is just draw a circle and say 4 millimeters at low power, okay? That's 4 millimeters. At medium power, again, would normally just measure this, okay? But at medium power, when we have 10 times 10, which 10 times 10 is not 1.6, so don't worry. It's because we're measuring it with a ruler. We're measuring the 1.6 millimeters, okay? So again, draw in the medium box this circle and say it's 1.6. Here's where it gets a little bit more difficult. Under high power, you can't use a ruler because there's nothing dividing up millimeters. So what we have to do instead is we have to do a little bit of a calculation to figure out field of view. So under high power, the field of view using a 10 times eyepiece, okay, and a 40 objective lens is 0.4, sorry, I'll go back, 0.4 millimeters. Don't write anything X, we're going to add something here. Because we can't use a ruler for high magnification, we're going to use this really long equation. You don't have to write this down. You're going to see it again here in a short second. So high power diameter divided by low power diameter equals our low power magnification over our high power magnification. It's essentially just two sides to an equation, high over low, low over high. So since we're looking for the high power diameter, we have to rearrange it. So what ends up happening is whenever you want to find high power diameter, so that length across your field of view, you're going to take your low power magnification and divide it by your high power magnification. Now reminding yourself, magnification means ocular times objective. So low power magnification is 10 times 4, and high power magnification is 10 times 40. And then we're going to multiply it by whatever our low power diameter was. So if we look at our last slide, in order to get 0.44 millimeters, we had to use this formula. So what did that look like? It looked like this. How did I get diameter? I took 40, divided it by 400. 40 times is my low power magnification. 400 is my high power magnification. Again, how did I find that? I just took my ocular lens times my objective lens. And then I figured out what that was, multiplied it by 4 millimeters, because if you look at your low power, that's how far the diameter was across. If you put that into your calculator, okay, you will end up with 0.4 millimeters, okay? And that's how I got 0.4 millimeters. I know this is probably like, okay, this is weird. So let's try a little bit of an example, all right? Oh shoot. So I gotta go get my pen. Where's my pen? Hmm. Oh, that's unusual. I must have gotten rid of it. Okay. Well, I'll just try and figure this out here in a second. Okay. So let's do it like this. Since I don't have my pen, I will type on it. So what you're going to do is in this example, and I believe you have this example in your notes, okay? We want to make sure to figure out how to get our field of view at our highest power. We're given our low power and we're given our diameter. So what we want to do is we want to ensure that we can figure out high power by going high power is equal to low power magnification divided by high power magnification. Multiply that by our low power diameter. When I put this into my equation, I'm going to get 50 divided by 250 times that by 3.7 millimeters. Okay, so that's my equation. In the end, I get 0.74 millimeters. So at high power, my field of view, if I'm looking through my microscope, the circle, the space that the circle is occupying is only 0.74 millimeters. Now this makes sense because as you use higher and higher objective lenses, as you get closer and closer with your magnification, your space is going to get smaller and smaller because you're trying to magnify those objects bigger. Okay? All right. So if you could add that into the example that is just below the chart. Fit number. So we're going to move on to another type of important part of magnification and microscopes, and that is the fit number. The fit number is the amount of organisms that fit across your field of view. Okay, it usually ends up being whatever is the longest dimension you're going to put it across. Again, this is just an approximate value. It's not perfect. Essentially, you're just going to guess how many fit across so you can use it for a calculation. So if I was to do this one, here is approximately how many fit across. Okay, so again, you're just making an estimate. So if I go back, you're looking at this organism here. How many times do you think it would have fit across? So looking, you can be like, well, it's this big, and you can kind of 4 or 5 times, okay? And it fits about 5 times. Some of you may have estimated 6, 7, but try to be realistic. It was big enough that you knew there wasn't going to be 10. It was small enough. Sorry, it was small enough you knew there wasn't going to be 10, but it wasn't too big that you knew it was only going to be 2 or 3. So give yourself a little bit of a leeway, 5 or 6 here. So then what you want to do is you want to use this to calculate um, the actual size of the organism. So how do you do this? Well, let's be honest. You have it covering the entire diameter. You have 5 of them. Your space is only 1.6. So you're going to take your 1.6 and divide it by 5. You want 5 equal sections. So take 1.6 and divide it by 5. So the actual organism is 0.32 millimeters. Now, all of this was leading up to just getting this written in your notes. So how do we actually find the actual size of an organism? Well, essentially, ladies and gentlemen, you take your field of view and you divide it by your fit number. Okay? And that's all you need to know. We will do practices of this, don't you worry. Now, the best part about bio is you get to sometimes draw the things you're looking at through a microscope. And we draw it to a size that we're comfortable with. So we put it on a piece of paper and we draw it so we can see all the structures. This is a problem because that's not actually the size it is. We're magnifying it on the piece of paper. We're looking through a microscope that's already magnifying it. Now you're magnifying it more. So what you want to do is you want to actually, when you draw it on your paper, measure the organism that you've drawn. So in our previous example, we knew that the organism itself was 0.32 millimeters. But now what we have done is we've drawn it on a piece of paper and it's now 11 centimeters in length, which is quite a significant size difference. We've magnified it quite a bit. So what we're going to do instead is we're going to now figure out how to do the magnification of a drawing. So all you do is you take your drawing size, making sure that your units are the same. So I measured it, it was 11 centimeters. I have to change that to millimeters in order to do this calculation because I can't divide centimeters and millimeters. Okay. So I changed it to 110, okay, because there's 100 millimeters in 1 centimeter. Sorry, that's a lie. There's 10 millimeters in 1 centimeter. I'm not sure where I'm going with that. And anyways, so 110 millimeters divided by 0.32. And what that does is that tells me that I magnified my drawing at 344 times. All right. So that is all of the magnification and everything about microscopes. There are some practice problems. We're going to do the magnification problems together. Then you're going to go off and do the extra practice ones in your notes. If you want to give the ones in your notes a try right away, the magnification problems to see how you're doing and then practice with me after, that would be great. I will talk to you soon and have a great night.